Oh, good afternoon, everyone. I'm Jonathan Patz. I'm the director of the Global Health Institute, and I'm pleased to welcome you to the 14th annual Global Health Symposium. I have the greatest honor and pleasure to introduce tonight's keynote speaker, Professor Susan Paskewitz. She's not only a professor, but she's chair of the Department of Entomology in the College of Agriculture and Life Sciences. And she's the co-chair for a new Midwest Center for Excellence for Vectorborne Diseases, which was funded by the Centers for Disease Control and Prevention at the tune of $10 million. Susan earned her doctorate in entomology from the University of Georgia at Athens and began her career at UW-Madison in 1991. She's at the forefront of addressing U.S. outbreaks of West Nile virus, Lyme disease, and she's also completed important work on malaria. So please join me in welcoming Professor Susan Paskewitz, our keynote speaker.
All right, great. Let me start by just saying thank you to Jonathan for extending the invitation to come and speak to the group tonight about my work and the work of many students and others on the problems related to diseases caused by ticks and mosquitoes in the world today. Before I really get going, I want to also thank the staff of the Global Health Institute. I had a really wonderful time interacting with Anne and Betsy and all the other folks who have to work together to put on an event like this, which is not trivial, let me tell you. So thanks to all of you for the hard work that you guys have done in putting this together for tonight. When I was thinking about what I wanted to talk about and how I would title this presentation, I decided to make it a little bit dramatic, and so I as you see, titled it Deadly Foes, Why Mosquitoes and Ticks Matter to Global Health and to You. I added that later. And you might think that that's hyperbole, that's maybe a little bit of overkill, but actually if you look at numbers about different kinds of ways that people die, what you'll see is that mosquitoes in fact do have an outsized, no pun intended, but an outsized contribution to human health. And so this number that you see here, 725,000 cases or people who die as a result of mosquitoes, is a dramatic decline over the last 20 to 30 years. And that's because we've made great progress on some fronts. Malaria, for example, where when I first started here, I would always tell my classes that malaria killed about 3 million people every year, and most of those were children under the age of 5. So, you know, from 3 million to 725,000 is a pretty good trajectory. So we made a lot of difference, but it's still, mosquitoes are really important. And they're not important necessarily because of the direct impacts, like they don't poison you when they feed on you. It's not that they inject a toxin that kills you. It's that when they feed on you, very often they have already fed on something else, and when they did that, they picked up one of these disease agents. And those can be parasites, bacteria, viruses, and all of those kinds of things can be transmitted by mosquitoes. Ticks also play an important role in transmitting these disease agents, both to humans as well as to oftentimes our pets, right, and our livestock. So I may slip tonight and start talking like a medical entomologist, and I might call ticks and mosquitoes vectors. So I just want you to know that that's what I mean by that term. You're a vector in the insect world if you can pick up a pathogen and move it around, you know, transmit it to a human or an animal. So I want to talk about the challenges that vector-borne diseases present us with. And so I'm going to share a little bit of information about kind of the breadth of different things that are out there, and then talk a little bit more about some of the things that have happened just in the last 10 to 20 years. And then I want to talk about this new center on campus and how we're going to work to try to develop some responses to vector-borne diseases. So probably many of you can think of some of these infectious agents right off the bat. So I told you malaria, that's going to be a very familiar one. Some of you may, if I say the word bubonic plague, right, you're going to know what that means. And you're probably going to be familiar with the system, the fleas that are involved in transmission of that kind of an agent. If you go down this list, though, you're going to see other things that maybe are not as familiar to you. And many of those parasites are quite rare. So these are things that you may never encounter, and very few people ever will. But some of them are unfamiliar to you because they actually occur in other parts of the world. And in those parts of the world, globally, they may actually be very important and very abundant. So I want to talk about, for the next few minutes, what happens when some of these global issues become local. And I want to talk about things that have happened, here in the United States, but also eventually I'm going to focus in on some of what happened right here in Wisconsin in relation to a couple of different invasions when new things showed up. And in particular, I'm going to reference two viruses, West Nile virus, and then I'm going to talk a little bit about Zika. So, way back in 1937, the West Nile virus was discovered in Uganda. And for quite a while, it was obscure. Many of us who teach medical entomology, unless maybe you were someone who focused on mosquito-transmitted viruses, you might not have ever even heard of it. It wasn't something that we knew caused a lot of illness in the world. In 1998, though, there was an outbreak in Israel, and it was probably that outbreak that resulted in the landing of this virus on our shores in 1999. So in 1999, the story was that in New York City, they started to see a number of elderly people showing up at their doctors, hospitals, and clinics with really serious neurological symptoms. And when you see a cluster of cases like that, where you have neurological disease, way more than you've seen in another year, so an outbreak, that immediately draws the eye and the attention of the state public health apparatus, and then the Centers for Disease Control will also be involved in trying to identify the source of this infection. Ultimately, in this first outbreak in New York City, there were 62 cases that were identified. 7 of those people died, which is a fairly high mortality rate for an infection like this. The CDC diagnosed this disease as St. Louis encephalitis, and that was something that we'd known about here in the United States for a very long time. It's a disease that would have kind of infrequent outbreaks or epidemics like this. So we thought we knew what was going on. But at the same time, at the Bronx Zoo, there was a veterinary pathologist named Tracy McNamara, and I was lucky to get to see Tracy present just a few weeks ago at the American Mosquito Control Association meetings. And she described what happened to her at this initial outbreak. She said what she noticed around the zoo was that there were a lot of dead crows. And she was a veterinary pathologist in charge of the health of the zoo animals. And so she was really worried about her birds, right? Things like flamingos, for example, exotic, but they're in the zoo so everybody can get a look at them. So she paid attention to the death of all these crows. And indeed, it wasn't very long before they did start having trouble. They had a cormorant die, some of their flamingos, they had a bald eagle that actually died. And when they began to look at the crows and the tissues from the birds, they realized that indeed it was not St. Louis encephalitis at all. it was a completely new thing that we had never seen on our shores before. It was this West Nile virus. So it took them 3 months from the original outbreak to be able to identify the right cause of that episode. And of course, then, everybody's curious, like, okay, how did it get here? We've never seen it before. How did it get to our shores? Well, eventually we figured out that outbreak had happened in Israel, and transportation. So I always ask my students in med-ed, what do you think happened to get that virus here? And most of them will say someone infected got on a plane and then got off in New York at the airport and then was able to infect, the local mosquitoes, which then infected the local birds and so on. That's actually not very likely because humans are a dead-end host for this particular virus. We can get sick, but the virus doesn't develop to high enough levels usually in our bloodstream for us to be able to infect local mosquitoes. So it's really the birds that are doing most of the infecting of the mosquitoes. So if it wasn't a person on a plane, then you have to think, well, how else could it get here? And people speculate it could have been exotic birds, that were brought in perhaps illegally. But my favorite hypothesis is that a mosquito got on a plane in Israel and got off the plane in New York City. And that has actually happened before. There are a number of cases where you can see people living around airports who got airport malaria, for example. So we know that mosquitoes do sometimes get on and off of planes, and it is possible that that's how it got here. That was one of our first questions. Of course, we also then were really curious about which mosquitoes are the important ones, so what's doing this transmission. We wanted to know, would the virus actually be able to make it through the winter? At that very first outbreak in 1999, people were thinking, well, maybe it's just going to be a one-off. We're going to have this one horrible outbreak, but it'll die out over the winter. We won't see it again the next spring, so that'll be great. Some people predicted that it would make it through the winter, but it would remain really focal in New York City, and it wouldn't become a bigger problem. Unfortunately, what we know now, after many years of West Nile experience in the United States, is that it does make it through the winter pretty much everywhere, and it didn't die out and become very focal in the New York City area. In fact, it spread across the country with great rapidity, possibly because birds are moving, migrating, and carrying it with them. It got here to Wisconsin in a bird in 2001, and then within the next few years rapidly spread across the rest of the country. So now every place in the United States has experienced problems with this particular virus. And what we know or speculate is that as many as 5 million people may have been exposed to this. Most people, when they get exposed, actually don't get sick. The vast majority tend to be asymptomatic. You don't even know you've been exposed. A small percentage of people might have fever, so they may feel more flu-like kinds of symptoms, but they get over it. And then a very small percentage of people actually have this neurological symptoms. We know now roughly 17,000 cases we speculate, of West Nile virus neuroinvasive kind of symptoms have happened. And so oftentimes when it's a neuroinvasive disease, those people never get to go home. They may be permanently impaired, and they will have often permanent symptoms of that infection. So a very serious outcome. I was just going to comment that another thing that we couldn't have predicted at the beginning of this invasion was this strange pattern that you see here, where eventually the hot spot for West Nile virus transmission turns out to be more in South Dakota than in New York City, which is not something at the beginning that anybody would have expected. We think it's because of the kind of mosquito that in that region is doing the transmission. It's a mosquito that's associated with agriculture and particularly with irrigation of agriculture, and can get to very high population levels. It also likes to feed on humans and birds about equally well. So it's good as a vector in that context. Well, after the initial invasion, we had three years where we had significant outbreaks. So one of those was in 2002. There was also a significant outbreak in other parts of the country in 2003, and another one in 2012. And notice the numbers here in Wisconsin, how things really declined from that first introduction in 2002 when we saw a big outburst of human cases. And we kind of thought, yeah, all right, so maybe it's going to go away, and that was it. But again, in 2012, we had another outbreak here. So I thought that I was beginning to understand, what was going on when we saw these kinds of bad years, and that it was associated with really hot and dry conditions. And I thought that that was because of something that I know about the mosquito. So the mosquito that transmits this is a mosquito that happens to like really stagnant, very fetid and smelly water. So the stinkiest water you can imagine, that's where this mosquito likes to lay its eggs. And so in a year when we don't get a lot of rain, the underground stormwater management system often really gets very full of water that's got a lot of rotting vegetation in it. It's particularly attractive, doesn't get flushed out by any rainstorms, and so these mosquito populations can be higher in those years. The hot also helps to make the virus develop a little faster in those insects. So I thought, okay, I got it. I got this figured out. But last year was actually the third highest year we've ever seen for West Nile cases in Wisconsin. And last year, I just pulled these off of the state climate office. Last year during the mosquito season, it was actually abnormally cool and abnormally wet, just the opposite of what I thought I understood about this particular circulation of this disease. So we still have a lot to learn. From this first outbreak in the US and here in Wisconsin, we learned a few things. We learned that our systems for detection and surveillance should be thinking a little bit more about incorporating people who work with animals, right? This One Health idea, that animal health and human health are often linked, and that we can use that in productive ways, in particular here for better detection and surveillance. I pointed out a number of things that were unpredictable. We didn't expect it to take root in South Dakota, so we just didn't know when something new comes in exactly what's going to happen. And then the last thing was we didn't have a vaccine, we don't have good treatment or drugs for this virus in people, so the thing we have to rely on is vector control, right? We really need to find ways to reduce the mosquito populations so that we can protect the human population. So now we can fast forward to 2016, and here our global transportation routes played a role again in moving yet another one of these mosquito-transmitted viruses into our own backyard. And this, of course, is one that I'm sure everybody in the room is very familiar with, and this Zika virus. And this virus probably showed up in 2014, 2015 in Latin America. By early 2016, it had been declared a global health emergency as a result of the epidemic levels of transmission that were happening in South America. And all of you know that we believe that outcomes of infection with Zika for a pregnant woman can be particularly bad. Very frequently, birth defects can result from that. And I was just reading something and spoke with someone who's the Zika coordinator for the state just a couple minutes ago about infections of newborn monkeys that also seems to indicate that you can have very negative impacts if you're not just a fetus, right, in the pregnant woman, but also for newborns. So as you might imagine, this caused tremendous consternation and worry all around the world. And unfortunately, it wasn't very long before people moving around again, going to vacation in Brazil or elsewhere, are bringing it back to our own shores. And in particular, down in Florida, in the Miami area, there was active transmission during 2016. And so, I remember during this period getting an email from the Centers for Disease Control actually asking me if I could take time off from work and go to Florida to help with the mosquito control efforts down there. And they really weren't talking about me like manning the con, you know, the emergency preparedness center and making sure everybody's getting their job done. No, they were looking for people who could be operational, you know, who could actually go out and help with the mosquito control efforts. And unfortunately, you know, those mosquito control efforts, what we have available is often the use of pesticides to knock down the mosquitoes. And here you see a guy using a sprayer, kind of going right up close around people's homes. And the reason for that is because this mosquito, very differently from the other one that I told you about that likes to breed in the nasty water, this mosquito is particularly adapted to human beings and containers around their homes. So we would describe this yellow fever mosquito, it's Aedes aegypti, we would call it peridomestic. It really likes to live in close association with people. It can use every little paper cup, a bit of trash in your yard that's got water in it to breed, lay its eggs. And to cap that off, it has a little bit different of behavior from many of the mosquitoes you're probably used to here in Wisconsin, which you're mostly going to encounter if you're going out, you know, once the sun is going down or when the sun is coming up, so more in those evening and early hours. This one is an aggressive feeder during the daytime. It really likes to come after you. There's a second mosquito you see here, the Asian tiger mosquito, that seems not to be playing as important a role in transmission of this particular virus, but we'll come back to that one a little bit later. Well, now, if you're a daytime feeder as a mosquito rather than one that's feeding at night, you know, and you're a guy working for a county and you're trying to figure out ways that you can suppress the population, you might well load up your pesticide in your truck and go out there and go up and down the road and spray your pesticide during the daytime, right? But what happens if you spray pesticide during the daytime for mosquito control is that you have these off-target kinds of impacts. And so this is one very negative outcome where someone who didn't have a lot of training, didn't quite know what he was doing, sprayed and killed millions of bees in this South Carolina focus. Nobody got any warning that he was going to spray. He did it when the bees were actively up and going around. And normally what people do is they spray in the evening hours when it's pretty quiet, so after the bees are not active. They put out this very tiny droplet fog, and in the morning, by the morning time, it's dissipated and it's degraded. So it does not have these kinds of impacts on the other kinds of insects. So this was a really unfortunate incident. And what it let us know, this outbreak, was that there are not enough people, medical entomologists, who can respond to these kinds of situations. We don't have adequate training for the people at the county and community levels who have to leap into action when you have an emergency like this and go out and try to protect communities. And the other thing we know and have known for a long time is that trying to control this particular mosquito, Aedes aegypti, is really hard. It's a very difficult thing to do because of its ability to use all those tiny spaces for laying its eggs. So I'm going to talk now a little bit about ticks. And I'm showing you a picture here of 3 different ticks. You're seeing the one on my right, the wood tick, which is really called the American dog tick, but everybody in Wisconsin calls it a wood tick, so am I going to call it a wood tick. The one in the middle is the Lone Star tick, and that's the one that's associated with the meat allergy, which thankfully is very uncommon here so far. And then the other one, which is our deer tick, also known as the black-legged tick, and then a tiny little nymphal stage, one of the immature stages of the deer tick. So what I want to talk about here is a little bit different from what we see with those mosquito-transmitted diseases, which are often like these big and dramatic episodes of an outbreak or an epidemic that gets everybody alarmed and worried and reacting, and then things kind of calm down. With ticks, we don't seem to have that same response. But we actually are seeing what I'd call a slow burn epidemic with every single disease that especially deer ticks transmit in the United States. So let me talk a little bit about that. Probably most of you know Wisconsin is a hot spot for Lyme disease. And here you can see a dot placed at every single county where there's a case of Lyme. And so Wisconsin and Minnesota, and then up the northern part of the eastern seaboard are places where Lyme is really common. So you all know that. The thing you might not know is that we're also a hot spot for emerging tick-borne pathogens. And so I'd show you a couple of things here. One, I always find it interesting to note that the first case of Lyme disease was actually reported from Wisconsin in 1969. It wasn't called Lyme disease at that time, but it was written up in a medical journal, and it was really clear from the way he described the symptoms, which was a bullseye rash, that's exactly what had happened. So Lyme was first here. We really didn't recognize that until about the middle 1970s, until the early 1980s, when the actual agent, Borrelia burgdorferi, was finally identified. And so there was a lot more surveillance for this particular tick and pathogen during that period. We first saw here in Wisconsin this human anaplasmosis caused by Anaplasma phagocytophilum in the early 1990s, showing up when a doctor at the Duluth Clinic recognized it in Wisconsin citizens and in Minnesota. And since then, that has grown dramatically. We have a lot more people infected with Anaplasma every year now, so probably 600-700 cases get reported to the state. We've just started to see some new things. So there's an ehrlichiosis. Another kind of Lyme disease is Borrelia mayonii. These are things that are still very rare, but they're definitely out and circulating. And then finally, this relapsing fever. So actually, if you look at the story with our deer tick in Wisconsin, is there are 7 different pathogens transmitted by this tick that can make people sick. And they include these bacteria that I'm just describing, but also Powassan virus, which was in the news a lot last year, can be a fatal infection. Again, a very, very rare pathogen and a rare infection in people, we think, but something that definitely is floating around here. And then this babesiosis, which is a malaria-like pathogen. And we've been seeing significant increases in Babesia as well. The interesting thing to me about that is that the hypothesis there is that Babesia is actually being facilitated by Lyme disease. And the story goes that when you have a mouse that's infected with Lyme, if it's also infected with Babesia, the Babesia levels in the blood can get much higher, and it makes it much easier then for that mouse to infect any larval ticks that are feeding on the mouse. So the two things seem to be related, this increase we're seeing with this disease. We're seeing, increases though with all of them. So why so many pathogens here? Why are we seeing so many new things? Well, I think partly it's because it's the whole Lyme disease and deer tick, it's a complex system. These ticks are generalists, and they can feed on lots of different animals and lots of different bird species, so they have a wide array of things that they could pick up pathogens from, and many of those, as we're seeing, are able to develop just fine in those animals. So I mentioned that it's a slow burn epidemic, and I put in so that you could see what that looks like. These are statistics from our Wisconsin Department of Health Services. And notice that back when I first started here in 1991, as Jonathan had to point out, I used to always tell my class there were 400 or 500 cases of Lyme in Wisconsin every year. But you notice that maybe right around 2000 there was an inflection point. And now what gets reported, or what we think is it's more like several thousand. Unfortunately, this is a little skewed because they changed the definition of how things get reported to the state. So some of this is extrapolation. But roughly, we're thinking 3,000 to 4,000 cases now for the reporting system. CDC says that's an underestimate, and maybe by as much as tenfold. So we could really be thinking 30 to 40,000 actual cases of Lyme disease in Wisconsin. So thank goodness, for the most part, it seems to be treatable if you see your doctor early. But all the other diseases, although we don't have quite 30 to 40,000 cases of anaplasma or Babesia, we're seeing these same kind of trajectories where year after year things are going up. So they're going up in part because we know that the tick populations are going up in many places. We're seeing more ticks, and we're also seeing higher infection rates in those ticks. We're also seeing changes in where the ticks occur in our state. And so these are a series of maps that show you surveys for where we find the ticks. And so the first one was in 1981, and the way this used to be done, we can't do this anymore, the way we used to do it was on the first day of gun deer season, the hunters would bring the deer to a registration station, and then we could have students at the station, and they would look at the side, usually around the face and the shoulder, and pick off every tick that they found in that area. That meant we could get a snapshot of where the ticks were in the state, across the whole state on one day, which is really valuable. And we did that a number of times. You could see that first year, you look in the whole eastern part of Wisconsin, you see any ticks on the animals there? So those pie charts, any place you're seeing a black, that's a percentage of deer that had at least one deer tick on it. So you don't see much. You go to 1994, now the center of the state, much more common then. And then in 2008 and 2009, moving over into the eastern part of the state. So over time, over these 20, 30 years, we've seen a real change in where these ticks occur. And that's contributing to those cases. I also wanted to show this because this was a summary of the whole region that was put together by a colleague of mine at CDC, Becky Eisen. And Becky is showing you kind of the same thing, like back in 1996. So that first map is showing you all the tick records from 1907 to 1996. And if it's red, we know the ticks are actively breeding, they're established in those places. If it's blue, that just means that maybe somebody found one tick one time, so we're not sure about those places. And you look at what happened, just between 96 and 2015, and I would say this is not even accurate. What I would say is any place in Wisconsin where there are woods, there are going to be deer ticks now. Every single place we go, if the habitat looks right, the ticks are there. So they have fully invaded all of the spaces. Where we tend not to find them, of course, is in like agricultural lands, right? So if it's a big, open, dry, sunny field, that's not the kind of place where these ticks can inhabit. They do like forested areas. Along with the movement of the ticks, of course, we saw the movement of the disease. And so this is just showing you that over the years. So why are we seeing this? This is not a static pattern, and it might seem a little bit strange, right? Like, why are the ticks increasing so much? And you might think, well, maybe it's the climate, something about that has changed. But I actually think this is probably more of a land use story. And it relates to the fact that in our history here, we had at one point pretty much deforested all of the landscape. So where there were trees, almost all of them got cut down. And similarly, most of the white-tailed deer, which are a really important source of blood for these adult ticks, most of the white-tailed deer were eliminated. It was market hunting, and a lot of that hunting was actually to feed the loggers in the camps, especially in northern Wisconsin. So you can see this estimate of what the white-tailed deer populations look like about 1900. And as both the forest and the deer have rebounded, there were probably small pockets of ticks around the state, just a few places. It took time, but eventually now they have been able to move out from those places and reinvade all of the habitat. So that's a little bit of a snapshot of some important stories for us. What's going to happen in the future? That was a bit of the past. What's going to happen in the future? I think we are going to continue to see sporadic West Nile epidemics. And I would have said before last year that if it was a hot, dry year, that's when we could begin to predict that we're going to have a problem. After last year, I have to go back to the modelers and see, what other things might we have missed. I think we're going to continue to see disease due to ticks and tick bite. Those are going to continue to increase as well. So those slow burn epidemics, those are not going away. And I think we're going to see new things coming into play. So we'll continue to have other kinds of mosquito-transmitted viruses imported into this country. What the next one's going to be, I don't know. Maybe chikungunya, maybe Mayaro, Rift Valley fever. There are a lot of possibilities. In addition to that, I think we're going to see more emerging pathogens coming out of our own tick-borne system here in Wisconsin. And so I'll talk about that in a minute. So along with that kind of unsettling, perhaps, view of what the future may look like for us in terms of vector-borne disease, there are going to be some positive things, some new things that are going to happen that maybe will help us to get a little bit better at responding. One thing, Tony Goldberg and some of his colleagues, for example, down in the Chicago area, are getting pretty good at being able to predict in advance when we may have a bad West Nile virus year. We can do that already, I think, in a temporal sense. So timing-wise, we know the piece we need to add to that is to know precisely what space is going to be bad so that we can really target our mosquito control into those areas where we think we're going to generate a lot of mosquitoes. So better predicting, better forecasting, that's going to be one tool that will help us. Another thing, really fascinating work that started a long time ago but was really kind of hitting its stride when I was in graduate school, was these ideas of thinking about ways to genetically engineer or change mosquitoes so that they can suppress their own population. So there's some really interesting work out there. A company called Oxitec that's been devising that solution. And then kind of along with that, that one has hit a little bit of headwind in terms of how the general public is responding to the idea of a genetically modified mosquito. But a parallel work has been to think about using microbes as a tool. And this idea is that there's an organism called Wolbachia. And some strains of Wolbachia, when you put them into mosquitoes, are actually able to, they can do two things. One, sometimes they can prevent that mosquito from getting infected with viruses. Wow, so now it's not a vector anymore. And two, they can also sometimes, different strains can be used, again, as a population suppression tool. So you grow them up in a lab in very large numbers, then you release them into the environment, they mate with the females that are out there, and none of the eggs or larvae that come from that mating are able to survive. And so they have one now called a company called Mosquito Mate. They got their Zap mosquito, which has this ability to suppress populations, and those have now been approved for release in 20 states in the US. So that is a tool that might be useful for Aedes aegypti suppression. So for the last little bit here, I want to talk about our new center and some of the things that we're also going to try to add to our ability to respond to disease outbreaks. So as Jonathan said, this is a regional center that was funded by the Centers for Disease Control. We have partners in 5 states, so we're working a lot with Minnesota, which has a very similar problem with ticks and tick-borne disease compared to Wisconsin. Also working a lot with Michigan, and so Michigan really hates us because the ticks that are now moving into Michigan are all coming from Wisconsin. Similarly, actually, I think the people up in Canada are blaming us for their situation with deer ticks too. And we also are working with folks in Illinois who don't have a big deer tick problem, but they have a lot of West Nile virus transmission, and they're beginning to see quite a lot of Rocky Mountain spotted fever in the southern part of Illinois, which is a little surprising actually to us. And then finally, we're also working with a lot of partners in Iowa, and I don't want to go on to my next slide before I make notice that the co-director of the Center of Excellence is Professor Lyric Bartholomew, who's in the School of Veterinary Medicine. And so she also has a wealth of expertise with mosquito and tick-transmitted disease. So the things that we need to do as a center and that we're really looking forward to making progress on are to try to do something about the fact that when we have these big outbreaks, we don't have enough medical entomologists who know anything and can deal with it, right? Like what has happened over the course of my career is I have watched all the funding for academics trend towards molecular biology. Right? So people who could understand how to actually go out into the field, collect mosquitoes, identify them to species. And by the way, we've got 60 different species here in Wisconsin, and they're not that easy to identify. So somebody's got to be able to do that, right? And somebody has to understand, what are the right ways to apply things and how do you protect the bees while you're doing it. So we're going to be working a lot to train the next generation. We're also doing some things to improve our ability to respond. And so let me just show you a little bit about what happened here when Zika virus, it kind of exploded onto the scene in the US. And at that time, a colleague of mine, Zip Johnson, was the Vectorborne disease coordinator downtown at the Department of Health Services. And Zip said, literally once this started, she could not get off the phone. Just hundreds of phone calls every day from worried people, like, a young couple wanting to take their honeymoon to South America and not knowing whether that was a safe thing, or a pregnant woman who'd come back, or somebody not pregnant but thinking about getting pregnant, like, how long are you protected, or what about the fact that Zika can be in semen, like how long do we have to use protection, and there were just a million questions that they had to respond to, and that meant that they dropped everything, right? And this is what they really had to focus on. One of the things that Zip didn't know how to talk to her community was whether there was any risk if somebody traveled someplace else and came back to Wisconsin, could some of our mosquitoes actually get infected from feeding on that person and then give it to other people in the community? And so that comes back to this question of which vectors and where are they? And the most important one being this Aedes aegypti mosquito, the yellow fever mosquito. You can see on the map here, and actually the distribution is much more restricted than what you're seeing here. This is a little bit of every state that ever had a record, is showing up as positive. So Aedes aegypti, I think there's very little chance of that one being an issue here. The other one, though, on the bottom, the Asian tiger mosquito, you see this really odd distribution where it kind of goes up into Minnesota but largely misses Wisconsin, and that seemed a bit strange to us. So we decided we would take a look just to see if we had this one. And so this was a little bit better map that came out in 2017 that showed all kinds of records from local mosquito control. So this might be an abatement district, for example, or maybe it's an environmental health group working for the county, collecting and identifying mosquitoes. And again, you can see from this map it looked pretty good, right? Like not too much in our area. And I'm giving you a close-up here, though, so look at this and see if you don't see something that seems a little bit strange, not necessarily in our area, but if you go a little bit south. Like, look at Indiana and look at Illinois. Does that make any sense? Well, to a medical entomologist, it does not make sense. The only way we can interpret that is that they're not doing the surveillance in Illinois. And so, we would think that perhaps the distribution is much wider in Illinois than what the map indicates. And if it's right on our border, then maybe there is an issue for us to think about. So if you want to know whether there are any mosquitoes of this particular type in Wisconsin, you don't put out stinky water for these. You do that if you're looking for West Nile virus. If you're looking for these mosquitoes, though, you can use the fact that they do like to lay their eggs in containers. And so luckily for us, you can buy stadium cups really cheaply. You can put your name and your phone number, have them print that on the cups for you. And then you can kind of pass them out to people who are willing to put them in their backyard with a little water in them and a tongue depressor. And then we train people to look at the tongue depressor and see whether or not there were eggs on it, and then ship them to us if there were, which we then hatched and identified. In the first year we did this, we had like 20 counties and lots of participants helping us. We found no Aedes albopictus at all. That was in 2016. The next year, we changed our strategy a little bit, and we started looking at places where tires were being brought into Wisconsin. And when we did that, actually, we and people in the surrounding states using the same kinds of strategy also found evidence of new infestations. So I do not think that this means that Aedes albopictus is breeding here. I don't think that this means that we have large populations or anything like that we'd have to worry about yet. I think possibly as it gets warmer here, though, or if we have little microclimates, might be right. We're testing that this winter to see if in the right kinds of protected places, they might actually be able to make it through the winter. So I don't think yet we have a huge risk from this Asian tiger mosquito, but I do want to point out that we have other things where our native mosquitoes, which are here in great abundance, as probably many of you know, are a concern. And here, an example would be last year, for the very first time, this Jamestown Canyon virus, which I would bet nobody out there, except for the people who work in my laboratory, have ever heard of, right? JCV, Jamestown Canyon. And yet, we had more than 40 cases last year in Wisconsin, and also similarly, what looked like an outbreak in Minnesota. We don't even know what the vector is. We have no idea which of our 60-odd species of mosquitoes is doing the transmission for this particular virus. And don't know what might have changed, so it suddenly seems to have emerged on the scene. The other thing that I find a little troubling is this Eastern equine encephalitis virus, which, as far as I know, we may have had one case in the last 50 years in Wisconsin. Last year, again, all of a sudden we had a lot of horses who were infected with EEE, and we had a human case and the person died. This is a really nasty virus. Well, this is not like most of them where the vast majority of people who get infected never get sick. EEE has a very high mortality rate, so we're really concerned about what's going on with that. Something else that our center wants to work on, in addition to surveillance and kind of understanding the vector relationships with these other viruses, is thinking about ways to protect public health from mosquito-borne disease outbreaks. And of course, you know, what entomologists do these days largely is try to figure out ways to kill bugs without damaging the environment. That's our job. And so we're always thinking about things like biological control. So could you use fish, for example, to eat the immatures or the larval stages in some kind of context? Or could you find a bacterial product that maybe just kills these mosquitoes but doesn't kill anything else? So part of what we're going to be working on for the center is to try to find some of these new methods to prevent disease. We're also going to work on ticks, of course. We have to do a lot of work on ticks because they're important to our own backyard. And so we're going to be working primarily on this deer tick, not on the wood ticks. As far as we know, wood ticks do not transmit disease in Wisconsin, although they're quite common. So if you're in the right places, you're definitely going to pick those up. And here is one of the people who works in the lab showing you the technique that we use to monitor the abundance of these ticks in our state. So one of the ways that you can do that is you go out and you use a piece of white fabric, which kind of mimics you, like walking through the woods and what might get on your pants as you brush up against the tick. And then we bring those all back to the lab and we can see, you know, whether or not they're infected with the different pathogens. This is really useful because some years we can see that we have big increases in tick populations and we can try to understand why, so then we can get back to our forecasting of risk. And we can also try to answer questions like, last year, for some reason, it seems like Lyme cases in Wisconsin might have gone up very dramatically. And we don't yet know whether that was a reporting issue or whether there really is something going on with the ticks. It does look like from some of our preliminary information, the infection rates in ticks were high last year, but we're still kind of looking at that data. And if it is true, you know, we're going to want to try to figure out why that might have been. We're also interested in tick control, just like we're interested in mosquito control. And so we'd like to be able to provide communities, neighborhoods, individuals, anyone who wants to be able to protect their own environment with evidence for what you could actually do, you know, what's likely to work. And so, of course, there's been a lot of work that's looked at spraying, putting pesticides in the yard. But we're very interested in this idea of trying to target the animals that are important in the cycle. So these little white-footed mice are particularly good at feeding ticks, and they're also particularly good at transmitting Lyme disease to those ticks. They're very good as reservoirs. So what can we do about that? How could we get the mouse into this control program that we're thinking about? Well, one way might be to think about treating the mouse much like you do your dog. So about now, I'm sure many of you are starting to think about this, and if you haven't yet, you should be, because the ticks are active. I already had one homeowner pluck a big one off a dog, send me a picture and say, what is this? Like, it's still snowy outside. But what do you do with your dogs, right? You go and you get stuff like Frontline, you put a little drip of that down the back of the dog, and then every time a tick or a flea gets on the animal, within a day or so, those are dead, so they can't reproduce and they can't transmit disease. The idea with these little tick tubes is we provide the mice with a nest material. They take it back, they build their nest with it, and it's got a little bit of an acaricide, an insecticide that'll kill a tick, incorporated into the fibers. And every time the mouse goes in, it gets a little dusting on the fur, and then every time the ticks try to attach to that mouse, then they're killed, just like the Frontline with your dog. And so we're testing this and have so far had some pretty good results in our project here in the UW Arboretum. We're also interested in some more out there kind of things. Again, thinking about whether one might be able to get at the problem by the mouse, but here vaccinating those mice. And we're not going to go out and catch every mouse and stick a needle in it, vaccinate it that way. Although one of my favorite colleagues actually did that one time just to see how it would work. She was testing an interesting hypothesis. So she trapped out mice in a large area like this. Every one of them got an injection, released them back into the environment, and then looked to see what that did to the rates of Borrelia in the mouse, but also in the tick population in that area. So really cool strategy. We have a different idea. We're going to put the vaccine into an oral bait formulation so that the mouse just has to eat it. So one of my students is currently working on that. Another one of the folks who's involved in the laboratory is really interested in how human behavior is important. And so now let me give you an idea. If you want to get involved as a citizen scientist, or if you want to help us understand the Lyme disease problem more, then keep an eye on this website. If you Google Wisconsin ticks, this will come up. And within the next month or so, we're going to have a link there to a survey or an app that you could actually download onto your phone. And then you'd be able to tell us when you get a tick on your person and what you were doing, which is really critical for us. We need to know how people are actually picking up the ticks so that we can do a better job of figuring out how we can educate and protect. So watch for the tick app if you're interested in helping us with some of our research. And then one of the other things we're really interested in is I mentioned to you that I think we're going to see more emerging infections in the state. And so we're very interested in this kind of One Health linkage to the animals that are really important. And here you're seeing a couple of students. I think one of them is actually in the room here today. And they're collecting white-footed mice and taking a little sample of tissue and also taking off all of the ticks that have attached. And then we bring those back to the laboratory, those and others that are just out looking for hosts, and we look at them to see what kinds of things might be in there that we didn't really even know were there and we never would be testing for. And so this is a really nice collaboration with a number of folks who are involved in the Global Health Institute. Tony Goldberg, for example, Christian Bernard, Lyric Bartholomew, myself, and then my student Avery Peace, who actually has a poster, if you're interested in this. And we're looking to see what kinds of viruses might be circulating in ticks that we didn't even know were out there. We've now found 5 things, 6 total, but we already knew about the Powassan virus, but 5 new things. And at this point, we don't know whether any of those might be involved in human illness. So I'm just going to finish by saying the other, wonderful thing that we get to do with our center is to work on that training goal. And so here are a number of my tick teams over the years who've done some great work for us in helping to survey the entire state of Wisconsin for ticks and small mammals. We get down and dirty and have to survive pretty terrible conditions sometimes. Like we're going to places where the mosquitoes you wouldn't believe. But they're a wonderful group. They get great training, great exposure to public health entomology, learn a lot of interesting things, and give as good as they get. So it's really wonderful to get to work with them. And I'm just going to finish here by thanking the funding agencies that have provided support for a lot of the work that we've done on these different issues, especially the Centers for Disease Control and Prevention. So thank you for your attention today, and I'll be glad to take some questions.
Perfect. Susan, thank you. That's an incredible presentation. So we have just a few minutes for questions. And while you're gathering your thoughts, be ready to raise your hand and we'll give you a microphone. We'll probably take 2 or 3 questions at once and you'll remember all of them and then we'll stop. But I want to take the prerogative of asking you one question, but first saying that one of the most important roles that you play is she's also on our advisory for the Global Health Institute. So we have some of the best people on our advisory giving us advice. Now here's a question I have. As you mentioned that Lyme disease is moving up into the UP of Michigan and they're upset about that, right? And you talk about land use change. There are some places like Sylvania Wilderness that have old growth forests with trees that are 200 and 400 years old. Are you seeing any differences in places like that compared to deforested places?
Yeah, that's a really interesting question, isn't it? And I'm in the same building with the Department of Forest and Wildlife Ecology, so there's some wonderful folks there who do a lot of work with old growth forest and contrasting those situations. And I think at one point we did actually think that there might be some differences, but we haven't, it's not something that we've been able to look at systematically. I think perhaps if that's a question that you guys would like to talk more about, we might be able to talk to Dr. Scott Larson, who's in the audience here, and who may have done some comparative work actually with some of those up around the Flambeau, was it, Scott? Oh, that's right. So one place we did go was the Menominee Indian Reservation because they're really famous for their deer management practices. They're really good at keeping the deer herd suppressed, and we were wondering whether that might also help to suppress the ticks. And they also have really excellent forestry practices, so they have a lot of interesting vegetation there that you don't see in some of the rest of the state. We found lots of ticks there, though. We were hopeful that it would be suppressive, but we didn't see that in that environment.
So we have, we'll just do one set, and then we're going to have to close. So.
Hi, thank you. If one of the tools you mentioned could wipe out the entire world's population of mosquitoes, would you support that?
So would I support a complete eradication of one particular species of mosquitoes? Because I would not support wiping out all of them. There are a lot of mosquitoes that don't feed on people. They maybe feed on other things. But let's say, just for the sake of argument, that we're talking about Aedes aegypti. So Aedes aegypti is an invasive species here. So I'm hedging a little bit. So I would be okay with eradicating it in the United States because it wasn't here originally, invaded probably 200 plus years ago. It's not native. Same with Aedes albopictus. I'd be okay with that. But if you ask me whether I would be comfortable with eliminating it in its native range, that makes me a little less confident. Some of my colleagues would say that if you look at the genomes of mosquitoes, if you got rid of Aedes aegypti, you'd be losing about 1% of genetic information. So basically, everything else that's in there is in other mosquitoes too. So you wouldn't be losing it at the genetic level. But nonetheless, it does make me uncomfortable, the idea of deliberately completely eradicating any species. That's because I'm a biologist, and actually I really love biology and diversity and all of the critters. You could probably tell I actually even like insects. Maybe not mosquitoes so much, but.
Speaking of diversity, we have a diverse number of great presentations upstairs. So please, we're going to stop with the questions now, and if you can help me thank Susan one more time. Now, I'd also be remiss.